Hello everyone and welcome back to another episode of Neurosci IQ. In this episode, we will be talking about postnatal development. Specifically, we will talk about some of the key events that happens inside of our nervous system. So let's get started now. Our first item on the agenda for today is myelin formation. If you don't know what myelin is, it is basically an insulating layer that forms around neurons, and this layer is typically made from proteins and lipids. It serves to reduce leakage and increase the speed of signal transduction. Actually, a fun fact is that myelin formation and demyelination are actually associated with many different disorders, and also some studies have suggested that myelin could also play a part in intelligence in humans and other mammals. Furthermore, myelin formation starts around the fifth fetal month of development, but the majority happens after we are born and continues until well after we are 20 years old. Myelin is formed by Schwann cells in the peripheral nervous system and oligodendrocytes in the central nervous system. If you don't know, central nervous system it basically compromises the brain and also the spinal cord. And as you can see, here is a diagram of a neuron surrounded by the myelin sheet that the Schwann cells are secreting. So now we will specifically talk about oligodendrocyte and how they mature and they develop the myelin layer in the central nervous system. Basically, this is a very complex and intricate process with multiple signals and which is tightly regulated. First, we have the oligodendrocyte progenitor cell. So this cell is basically, it hasn't received any signals yet to become a mature oligodendrocyte. After it receives a few key signals, it becomes an oligodendrocyte precursor. So now it is committing to the pathway of becoming a mature oligodendrocyte. The next key step here is that it becomes a pre-myelinating oligodendrocyte, and this is a very key step for oligodendrocyte as it is the final checkpoint until they become their mature myelinating form. After they become mature and they are now called the myelinating oligodendrocyte, they start associating with neurons' axons and they start forming the myelin sheet around the axons to insulate the neurons. One thing that is very important to know is that actually mature oligodendrocytes are a heterogeneous population of cells, and what this means is that they have basically different transcriptional profiles, and for example we could have a different oligodendrocyte in one part of our brain and another oligodendrocyte with different transcriptional factors and proteins in another part of the brain. Another thing to remember is that this process is very tightly regulated with many different steps and signals. Actually, 50% of immature oligodendrocytes undergo apoptosis and basically they die. It is still unclear about the specific mechanism and the pathways, but the BAX pathway and the BAK signaling pathways are thought to be very likely to be involved in the apoptosis of these cells. Furthermore, as I mentioned, oligodendrocytes and myelin formation are involved with many different diseases. One disease that is specifically involved that we should know is autism. And again, we can also see the neurons and how the mature myelinating oligodendrocyte has insulated the neurons. And the white part represents the myelin sheet that's insulating the neuron. So now, just before we continue until the next part, I thought I should clear up some very common misconceptions. A lot of people think that the brain volume, as it increases from when we are a baby to adulthood, that this is due to neurogenesis or the formation of new neurons. However, this increase in blood volume is not due to the formation of new neurons, and actually we form very little or even none in terms of neuron formation. This increase in brain size is actually due to one, axon myelination, as we learned, and also dendritic arborization, which basically means the dendrites mature and become longer and more intricate, and also synaptogenesis. So again, the brain volume increases not because of new neurons being formed, by other processes such as the myelination of axons. So now let's talk about another very important process that largely happens after we are born. What happens is the synapses that we made during the fetal months and also during early development actually get removed. But don't worry, because this is a very normal process and it happens all the way until late 20s and in some cases even after that. And a good principle to remember this by is the use it or lose it principle. And what happens here is that if we are not using a set of neurons, then the synapses eventually get removed as we are not using the neurons, and we need to dedicate or allocate the energy and the resources towards other brain functions and also strengthening other parts of or other neurons and other synapses instead of just keeping these useless synapses that we don't use. If you're wondering about the mechanism, one of the common mechanisms that the scientists have figured out is actually the mGlu1 basically stimulates the expression of the chemical semaphorin or SEMA7A, and the release, and this basically stimulates the release of BDNF from the Purkinje neurons, which facilitates the synapse elimination in neurons. And what happens is that when we don't use the synapse for for example, a period of time, these chemicals get released and they tell the immune system, for example, microglial cells, to remove the synapse from the brain, and basically the immune cells engulf the synapse and remove it. Conversely, if we are using a synapse and it is very useful and we are using it very frequently, the Purkinje neurons release SEMA3A factor, and this is a strengthening factor, and it promotes maturation and stabilization of the synapses that are regularly used. So in summary, the synapses that we don't use get eliminated, so we are not wasting excess energy on them, and also the synapses that we use frequently get strengthened, so we are making sure that we are not eliminating those that we need for later on. So now let's talk about neuron plasticity. The key thing to remember here, as many of you guys already know, is long-term potentiation. And what happens is long-term potentiation is actually a very widely studied phenomenon in the brain, and it is seen in many parts of the nervous system, such as the cortex and even the spinal cord. And to briefly describe what long-term potentiation is for those of you that don't know, is it is a persistent increase in signal strength between two neurons. So basically what happens is if one neuron keeps on firing and activating another neuron, is eventually the brain or the body recognizes that these neurons are activating each other frequently, and what happens is the next time that neuron fires, it actually makes it easier to activate the other neuron. And what happens in basically scientific term is that LTP or long-term potentiation is basically associated with an increase in the EPSP amplitude. And also, as I mentioned, it is very widely studied because it is implicated in memory formation and learning. So when we are learning a new information or a new like an activity, the neurons in our brain are forming long-term potentiation, so we can organize the neurons in a way that is beneficial and we can learn or create new memories more efficiently. So now we will cover a type of long-term potentiation that is very commonly studied. And this LTP is dependent on NMDA receptors. And if you look at the diagram, basically what happens is glutamate is released onto the synapse, and the glutamate activates AMPA receptors, which leads to the depolarization. After this wave of depolarization, if it's repeated multiple times and if it's strong enough, then eventually what happens is the magnesium ion that was blocking the NMDA receptor gets removed from the NMDA receptor, and the calcium ions start rushing inside the cells via the NMDA receptors. And what happens is eventually this influx of calcium ions actually start or initiate a cascade of events inside the cell that leads to the nucleus to transcribe more proteins and other transcription factors. And the downstream effect, long story short, is that we create additional AMPA receptors, so the neuron becomes even more sensitive to glutamate release in the future. So thank you so much for watching. If you enjoyed this video, please make sure to like and subscribe for more similar content in the future.